Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on the structure and functions of viruses. In our last lesson, we discussed recombinant DNA. We discussed the role of bacteria in producing recombinant DNA, and we looked at the stages in the process of genetic modification. Today, we will shift our focus from bacteria to viruses. We will learn the structure and function of viruses, and we will look at a few examples of this microorganism. Are you ready to begin, students? A virus is a small infectious microorganism that can replicate inside the living cells of organisms. Most viruses are too small to be seen directly with a light microscope. In fact, viruses are much smaller than even the smallest bacterium. Viruses can infect all types of organisms, from people to plants. In our previous grades, we have learned that there are reasons to not consider viruses to be living organisms. A virus particle, or a virion, is nothing like a prokaryotic cell or a eukaryotic cell. There are four main characteristics of viruses. The first characteristic is their extremely small size. Most viruses are between 0.01 mu meters in length. This is 1,000 times smaller than the smallest bacterium and 1 million times smaller than the average human cell. The second characteristic is that viruses do not have a nucleus. The third characteristic is that only some viruses contain a small amount of DNA, while others only contain RNA, but no chromosomes. The fourth characteristic is that viruses do not have any organelles. Since organelles are not preset, viruses cannot carry out normal cell activities such as respiration, active transport, or facilitated diffusion. Students, let us do an activity to review what we have just learned. Please determine if the following statements are true or false.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. I am sure this activity was simple for you. Let us review the answers together. The statement, "Viruses are 1,000 times smaller than normal human cells," is false. The statement, "Viruses can only infect people," is also false. Viruses can infect all living organisms. The statement, "Viruses do not have a nucleus," is true. The statement, "Viruses cannot perform photosynthesis," is also true. Since viruses do not have organelles, they cannot perform metabolic processes that take place in other cells. The statement, "A virion is a virus particle," is true. The last statement, "Viruses do not contain genetic material," is false. Viruses contain small amounts of DNA and some contain RNA. How did you do? Great work so far, students. Let us look at the structure of a typical virus particle. A virion consists of 2 or 3 parts. As you can see, the genetic material made from DNA or RNA is located in the center. It is surrounded by a protein coat that protects these genes. An envelope of lipids surrounds the protein coat. This envelope is a membrane from a host cell. It is important to note that not all virus particles have an envelope surrounding the protein coat. The shapes of viruses range from simple helix forms to more complex structures. Another characteristic of viruses that varies is the way in which they infect cells. Students, for our next activity, draw a virion on a piece of paper. When you have completed your drawing, label the parts of the virion that we discussed today.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, students. I am sure everyone was able to complete this activity. Your drawings should look similar to this. You should have labeled the genetic material, DNA, or RNA, on virion. As well, the protein coat and the envelope of lipids. Great job, students! Viruses are parasites. This means that the only way they can reproduce is by invading other living cells. Viruses take over the metabolic process of the cells they invade and make the cells produce more virus particles. Once the virus has reproduced in an organism, the newly formed virus can escape and go on to infect other cells. Let us look at how two different viruses infect a cell. This is an enveloped DNA virus attaching to the cell. Once it enters the cell, genetic material of the virus enters the nucleus of the cell. Then the genes replicate. After genes are replicated, the virus reassembles and escapes through the host cell's membrane. Now, a new viral DNA particle is released, which can go on to infect other organisms. When a DNA virus infects a cell, viral DNA replicates and controls the synthesis of virus proteins. This means that new DNA and proteins can be assembled into virus particles. Next, we will look at how non-enveloped viruses infect a cell. The RNA virus enters the cell, removes its coat, and replicates within the cell. Then, it reassembles and breaks through the cell membrane. A viral RNA particle is released, which can then go on to infect other organisms. When an RNA virus infects a cell, it synthesizes more viral proteins, including more RNA proteins. The new RNA proteins are assembled into new virus particles as well. We have reached the end of our lesson. Today, we began our study on viruses. We learned how viruses are structured and their main characteristics. We also learned the functions of viruses and how they infect cells. Until next time, thank you, teacher. Thank you, students.